Welcome back to the EDGE tutorial series. This video will go into detail for the pre-processing module in EDGE. After this tutorial, you will know how to set up parameters for quality trimming and filtering. Additionally, you'll know how to remove host genomes from your data if necessary. Keep in mind that the EDGE workflow is designed for Illumina data. We have limited capabilities for PacBio and Nanopore. Feel free to contact our dev team with questions regarding data from other platforms. As a final reminder, we are running two job submissions in parallel. The first uses the E. coli FASTQ files in the PublicData testData directory. The second uses data from the SRA runs SRR 2164314. The pre-processing module has two components. First, we will go over quality control. The quality of sequencing reads is never perfect, and read quality often declines as the read is extended. You can see an example of this pattern in the upper left box plot here. The quality of raw reads on the y-axis is plotted against the input read position on the x-axis. You can see how the quality scores get worse and are more variable the longer the read. The quality trim and filtering module in EDGE uses software to rapidly evaluate quality and remove unwanted reads. By default, this module is turned on. For the majority of cases, the default parameters are appropriate to filter out poor quality reads. Exceptions can arise when specialized adapter sequences have been added. In this case, a user can supply FASTA files or specify the number of base pairs to trim from each end of the reads. After quality filtering and trimming is complete, poor quality reads are discarded. You can see these differences by looking at the graphs for input reads versus trimmed reads here on the right. The quality here is much higher. You can also see in this bottom graph that these poor quality reads circled are no longer here. The second component of the pre-processing module is the host removal feature. This tool is used to subtract unwanted reads that align to a selected reference. Unwanted reads derived from hosts or positive controls like phi-X are easily filtered out in this step. For example, human reads are easily removed from clinical blood samples when this module is applied. At the EDGE interface, this module must be turned on. Users can select reference genomes available within the EDGE software or upload their own FASTA file containing reference genome information. 90% similarity is the default and lowest recommended setting, although this is left up to the user's discretion. Although highly recommended, pre-processing is not a requirement for downstream modules. Let's go to the EDGE interface and update our pre-processing settings. First, let's look at our E. coli sample. You can see how the FASTQ files are set up. Scroll down to the pre-processing module and click for the drop-down options. Under A, Quality Trim and Filter, we will leave all the parameters at their default settings. Again, these settings have been designed to be robust and should work on the majority of samples. If the quality looks really funky, users can go back and toggle these parameters on a case-by-case basis. Now let's click on B, Host Removal. Remember, we will need to change this setting to Yes in order to run host removal. For the host genome, we are going to go into the drop-down menu and select phi-X. The selected genomes here will depend on each sample origin, and it's up to the user to determine. Click on the X here to save your selection. Okay, let's update the pre-processing settings for the SRA data. We are using the SRA data SRR 2164314. Scroll down to the pre-processing drop-down menu. Once again, we are going to leave all of the quality trim and filter as is. Click on host removal. We want to turn this on. Yes. For host removal, we will select the human reference because we know this sample originated from human feces. We will also go ahead and select Phi-X to remove any positive controls in the sample. Click X to save our selection. That's it for the pre-processing module. Next up, we will dive into assembly and annotation.